large molecule, and this is our big guns, right? We're never gonna start with vancomycin, right? We're gonna start with our penicillin, and we're gonna work our way down until your last resort. Okay, nothing is working. We're gonna give vancomycin, right? And that's gonna be because we want to limit the exposure that we give to bacteria of vancomycin, right? Every time you show it, you're creating the potential to have drug resistance, right? We already have bursa, right, which is intermediate. Visa, right, which is intermediate. Staph aureus. Okay. We talked about at the bottom, it's isoniazid, right? Isoniazid is an antibiotic that specifically targets atypical bacteria or mycobacterium, aka bacteria that have mycolic acid cell wall. Okay, so this is an antibiotic is it narrow or broad? Narrow, because it specifically targets mycobacterium. Okay. Vancomycin does work best on gram-positives because they're the ones that have that thick layer of peptidoglycan exposed, right? That doesn't mean that you can't use it on a gram-negative. If you want to use it on a gram-negative, you're going to need probably a higher dose or give it intravenously, okay? But it works best with gram-positives. But we can try to manipulate to make it work. And what you'll learn too when we talk about antibiotics going forward is we do have certain antibiotics that we give in specific cases, but sometimes we have to manipulate when it's best to give one over the other, and it's mainly a case-by-case basis, right? It depends on your patient. Okay. Questions on cell wall synthesis. So if you're making this your summary slide, I would add in this section the ones that are on your worksheet, right? If you want to have a visual to go with your worksheet, I would just add the ones that we talked about right here, okay? So you don't forget them. Okay, so now we're gonna move on to the second mode of action, okay? Disrupting the cell membrane. So first, antibiotics that disrupt the cell membrane, remember PA, right? Some of you may want to become a PA. I don't know, but PA is going to be polymyxins and amphotericin. Okay, that's your PA. That's on your list. Okay, so what these guys do, so polymyxins, these are molecules that can insert themselves into the cell membrane, and basically they create pores. If you create pores, then you mess with the cell membrane integrity, right? Now other things can come in or leave the cell that normally shouldn't be able to. What does that lead to? Cell death. Okay. Polymyxins can be a little bit more toxic because as we look at some antibiotics, if they target something that we have as well, then there's going to be or can be potential for side effects, right? Before, when we talked about our illins or enums, cephalosporins, these guys are all targeting peptidoglycan, right? Do we have peptidoglycan? No, so we don't have to worry about that when we think about selective toxicity, right? But with polymyxins, we have cell membranes, right? So can our cell membranes potentially be affected? Yeah. Okay. So this probably isn't our first choice, okay? But these are gonna work well not with gram-negative bacteria. Why gram-negatives? Because remember, gram-negatives have an outer cell membrane, okay? So we can target that outer cell membrane, okay? They're gonna insert themselves inside, create these holes, okay? So it acts kind of like a detergent, okay? Similar to when we poke holes with Dawn dish soap into our E. coli, same concept. So it's gonna, here is amphotericin B, okay? This guy, it has a polar side and a non-polar side, okay? So it can insert into that cell membrane and create this pore. Things can leave and come in that shouldn't be able to, okay? Which leads to cell death. Okay? So this is something that is bactericidal, right? Kills the cell. Questions? Okay, that's it for cell membrane. Okay. Now we're going to move on to the inhibition of protein synthesis. Okay. So remember, polymyxin, don't forget to add amphotericin to it. And if you're making this your summer slide. Okay. So inhibition of protein synthesis, there's 4 that you need to know. Okay. You do need to know where they specifically target within protein synthesis. So I'm gonna start with chloramphenicol. Okay, chloramphenicol has a P in it. Okay, not to be confused with cephalosporin, because that may happen. Like if your option is chloramphenicol and cephalosporin, and you're like, God damn it, which one was which? Like, don't forget that one. That's like a common, like, you know how 2 things always look really good? Those 2 are gonna look really good when you see them together, okay? So, chloramphenicol has a P, okay? That P, think of peptide bond. So, chloramphenicol targets the peptide bond that is being formed between this amino acid and this amino acid. So, if we can't form a peptide bond between this amino acid and this amino acid, can we ever make a protein? No, because you can't link your amino acids together. Okay. Tetracycline. Tetracycline starts with a T. Okay. Tetracycline targets the tRNA. Okay. tRNA. Okay, how does it do that? It prevents the tRNA, that anticodon region that's supposed to bind with messenger RNA. It prevents that from happening. That recognition is not going to happen. So tetracycline attacks tRNA. If your tRNA can't link up with your messenger RNA, can you put in the correct amino acid? No. If you can't bring in the amino acid, can you make a protein? No. So protein synthesis overall, but where specifically? The tRNA. Okay. Streptomycin. Okay, streptomycin, think of S. Okay, starts with the letter S, and it targets the 30 subunit, the 30S. Okay, SS. So what it's happening in the 30S region is it creates this kind of snake, right? S kind of looks like a snake, right? So it creates this bubble in the messenger RNA. By creating a bubble in the messenger RNA, you can't read the sequence correctly. If you can't read the messenger RNA correctly, how do you know which protein to even bring in? You don't, right? If you can't read the sequence, can you add an amino acid? No. If you can't add an amino acid, can you make a protein? No. So all 3 are targeting protein synthesis, but in different pieces, okay? Chloramphenicol, P, peptide bond. Okay. Tetracycline, T, the tRNA. Okay. Cephalosporin, S, creates an S, bubble, 30S, part. You like it? It helps. Okay. Okay. Aminoglycosides. Okay, aminoglycosides, these are going to be streptomycin. Okay. Aminoglycosides, what they do is they kind of have bad side effects. Why? Because it's targeting messenger RNA, okay? We got messenger RNA too. So can it affect our messenger RNA? Yeah. So whenever we give like streptomycin, one of the bad side effects is gonna be things like kidney damage, okay? Kidney damage, and then your last, when it talks about targeting protein synthesis, and it starts with the G, that's gentamicin, okay? Gentamicin and streptomycin are aminoglycosides, okay? So in that worksheet level where it says which one of these are aminoglycosides, it's streptomycin and gentamicin. And then what happens, because they are toxic, we give them in a smaller dose, okay? If you give some of these antibiotics, like these for example, when you give at a lower dose, you're gonna have more bacteriostatic effects instead of bactericidal. Okay, here are some synergistic examples. So I remember in lab we talked about, we kind of talked about this, but we hadn't learned about it yet. But these are antibiotics that work synergistically, are going to be antibiotics that work better together. Okay, beautiful. They work better together, right? When you look at, you have like that extra added layer, right, on the top and the bottom, and that made like our cute little smiley face. Okay. So examples are going to be gentamicin with the penicillin, right? Penicillin is going to break the cell wall. Gentamicin targets the ribosome. So guess what? You're exposing the cell wall. Gentamicin can come in, right? So this is kind of hitting with like, this is the same as if you get hit in the face and then you get like uppercut to the chin, right? You're targeting the same spot. They work, your person's gonna go down a lot easier, right? Hit from the left and then uppercut, boom. Okay. Another example is gentamicin and carboxylin, okay? What do these guys have in common? You have a ribosome / protein synthesis, and the other one is a cell wall synthesis, okay? So you can say any type of ilin, enum, ceph mixed with something that targets protein synthesis, right? Except for tetracycline, right? Remember we said not tetracycline? Do you guys remember why not tetracycline? With the penicillin? Yes, tetracycline is going to be a bacteriostatic, okay? If you're giving penicillin, penicillin works best on a cell that's actively dividing. And tetracycline stops, right? It prevents replication, right? It pauses the bacterium. If you're pausing the bacterium and then you give penicillin, then what's the point? It inactivates penicillin. So you never want to give that combo. Okay. Tetracycline, okay? Tetracycline can target gram-negative, gram-positives, and then intracellular organisms. Tetracycline is broad or narrow? Broad, okay. So that's great, okay. But tetracycline, when we think about in its specific category, this is the last thing we want to give, okay? Because it can be very toxic, okay? Tetracycline, okay. It's bacteriostatic, so it's not a good synergistic with penicillin. So if I say, what is something you never want to give together? Penicillin, tetracycline, okay? Because tetracycline can kill almost anything, it's going to kill a lot of your good flora, which is bad, right? But if you have a really bad infection, right? It can kill Pseudomonas, right? As we saw on your plates, which one was the hardest one to kill? When we're looking at disinfectants. Yes, so we can kill Pseudomonas with tetracycline. So it's a good antibiotic, but there's a lot of, I'll talk about a little bit more bad side effects to it. But it works really well against Pseudomonas, right? It works with Proteus, okay, and then Staphylococcus bacteria, okay? But again, it can cause things like kidney damage, nausea, diarrhea, liver failure, and just a general rule, when you're taking antibiotics and birth control, antibiotics does interfere with the mechanism of birth control. So if you are taking antibiotics, then you need a different, like a second contraceptive, because a lot of people, if you're going in and you're taking your antibiotics, and you're on birth control, right, the pills, they like inactivate it. So if you care to not be pregnant, you need something else, a secondary form of contraceptive. Tetracycline, think teeth. Okay. Tetracycline, it binds to calcium. Okay. Where do you have calcium? In your teeth and in your bones. So it can cause tooth decay and brittle bones. Okay. Also, if you are pregnant and you take tetracycline, who else has bones? Baby. Okay, so that can cause deformalities. Okay, so you never want to take tetracycline if you are pregnant. There's a lot of antibiotics that you typically can't take when you're pregnant, but definitely not this one. Okay. Questions? Okay, next one, chloramphenicol, right? P, which means we're attacking the peptide bond. Okay, chloramphenicol, okay. Chloramphenicol is completely synthesized in the laboratory. Okay. Ta-da, we came up with that. It's a broad spectrum. It's bacteriostatic. We typically use it to cure things like typhoid fever. You don't need to know that chloramphenicol treats typhoid fever and severe rickettsia. I do care more that you know what chloramphenicol targets, right? What is the mode of action? And that is broad spectrum. It is bacteriostatic. Okay. Side effects to chloramphenicol. It can cause reversible aplastic anemia and permanent aplastic anemia. So with reversible aplastic anemia, so chloramphenicol, right, it targets that peptide bond, so that's protein synthesis. Basically what happens is you can't produce as many red blood cells. Okay? You can't produce as many red blood cells, that's gonna be a problem, right? You also can't produce as many leukocytes, right, platelets, so it messes with your bone marrow, okay? With reversible aplastic anemia, you stop taking the antibiotic, and then your body can just function normally, okay? So just take it away, and then everything's fine. With permanent aplastic anemia, basically, you're gonna need a bone marrow transplant. So that bone marrow, right, which is responsible for producing red blood cells, leukocytes, platelets, all that, that is now damaged, and you need a complete transplant. If you don't get a transplant, you'll probably die. Okay. So probably not the first thing we want to give. Okay. It's not very common though. Look, 1 in 2.5 million, right? So It doesn't happen very often, but it can happen. Yes.
Does it have to do with how long somebody has been on it, or just how their body reacts to it?
I want to say it's how their body reacts. So, and how immune compromised the person is. Are they old? Are they young? Right? Yeah. All of these side effects are going to be worse depending on your patient's condition. So when you're thinking about your case study, right? And we talk about what antibiotic you need to give, right? You can tell me potential side effects, right? Depending on how bad your patient is though, you're just focused on killing the bacteria, right? Because if you can't kill the bacteria, you're gonna get septicemia and you're gonna die for sure. So you're just like, let me target what's in front of me right now, and then we'll worry about the rest later, right? That's why you're like, why even give tetracycline if it does all that? Because we're worried about the bacteria in that moment, right? Once we target the bacteria, we'll deal with everything else, right? We'll deal with the teeth later, right? Or if you're in a position where, yes, you're pregnant, but let's say you're in a position where you're severely compromised and like you have to choose like who's priority saving. If your priority is saving mom at that moment, you're gonna have to go with the tetracycline, right? Because if the bacteria takes over, right, septicemia, you're gonna be dead. So they're just like, let's clear this bacteria however we can. So it's kind of one of those, right? So not first choice. Depends how bad your patient is. Okay. Yes.
Is tetracycline the same as a doxycycline?
Not 100% sure they're exactly the same.
Because I've heard doxy being used for like for resistant STI. So this is the first time first time I've heard about tetracycline.
It's probably a derivative of tetracycline because they have the same ending. So I want to say it's probably a derivative of it. I'll double check, but I'm pretty sure it's a derivative. Okay, those are inhibition of protein synthesis. Okay, now we're going to talk about, I think we're going to talk about the metabolites first, and then we're going to talk about inhibition of protein synthesis, or this one or that one. I don't remember which slide comes first. Okay, whichever slide comes first, we'll talk about it. Alright, there we go, it was the metabolites. I'm like, I feel like it's metabolites, but it says 4-5. Okay, so metabolites, right? So we're targeting a metabolic pathway, right? Metabolic pathways, who is in charge of driving these reactions? Just like in general, like what drives a reaction? What makes a reaction go? An enzyme, right? So we're targeting a lot of times enzymes, right? When we talked about heavy metals, how a lot of times heavy metals can inactivate certain proteins, right? So we're targeting enzymes that are involved in key metabolic processes, okay? Because if we block a key metabolic process, we can kill the bacteria, okay? So we can use these for like protozoans, different fungi, bacteria, viruses. So for example, we've already talked about sulfa, right? Sulfa is an antibiotic that is going to bind competitively where PABA is supposed to bind. Okay? If it binds where PABA is supposed to bind, then PABA can no longer bind to that enzyme. Okay? If PABA can't bind to that enzyme, you can't make folic acid. Okay? Folic acid is key. Okay? If you can't produce folic acid, your bacteria can't replicate. Okay? So bad, right? Okay. Sulfa is 100% synthetic. So we just made this up. Okay, we took chemical property and knowledge, and we just made that up. There is no organism that produces sulfa. We just made that in the lab. Okay. It's bacteriostatic, typically, right? Can be, it depends. Okay. So the two that you should know is sulfanilamide, which is part of the folic acid synthesis, and I'm not gonna hold you to clotrimethazole. I care about trimethoprim, okay? Trimethoprim and sulfanilamide, okay? Those are the two I care about. These two guys, okay, in that column, when you give them together, they work better, okay? If you give two that work better together, is that going to be synergistic, additive, or
Synergistic.
antagonistic? It's synergistic, okay? So the reason why these are synergistic is because they both target two different enzymes within the whole folic acid mechanism. Okay, so there's the first enzyme involved, it's sulfa, and then the second enzyme, that's trimethoprim. So because they're both targeting the same pathway, it's synergistic, right? This is like socking someone in the face from the left and then uppercutting the chin, right? They're targeting the same pathway. Okay? No. Yeah, that's not even on the board. That you don't need to know that. But they are synergistic. Okay. If something is additive, that means they work well independently, right? That's what additive means. So that's like you hit someone in the face and then you kick them in the knee or you kick them in the ankle, right? They're going down, but you're targeting different areas, okay? Not the same spot. Okay. The reason why we would want to give something that's additive, so even though they don't target the same specific area, they're still bringing the organism down, and by doing additive, you can reduce multi-drug resistance, and you can reduce the dosage that you're giving of each, so less toxic for your patient. Okay? So those are the only two for anti-metabolites. Okay, now we're going to talk about targeting nucleic acid synthesis. So this can either be DNA or RNA. Okay? DNA and RNA are both nucleic acid. Okay. Oh, that's blurry. Okay. So we're going to talk about targeting transcription. Right, transcription is making RNA from DNA, and we're going to talk about replication, right? Targeting making more DNA from DNA. So nucleotide-based analogs. Okay, these are going to be drugs that look like a nucleotide, but they're not a nucleotide, okay? The way we design them is we introduce them hoping that they get introduced as a nucleotide. But if you have an incorrect nucleotide get inserted, that's gonna mess with, right, the shape of the DNA, right? If we mess with the shape of DNA, then you can't replicate it, you can't transcribe it. Right? This is going to be really, really effective against organisms that are actively dividing, right? Make that log phase. These are effective against cancer cells, right? Because they're dividing really, really rapidly. okay? They're dividing so rapidly that they're not really spending a lot of time in this proofreading area, right? Because it's really, really fast division. These are really effective against viruses because viruses don't do proofreading. Yeah. Okay, so here's what they look like. Okay, here we have thymidine, right? Thymidine is a part of DNA or RNA? DNA, okay? You can also tell because it has the hydroxyl group right here, right? Okay. Here, looks like thymine. Is this thymine though? No. This is AZT, okay? AZT or, AZT, I'll call it AZT, okay? AZT looks like this, but what's the difference? Yup, no hydroxyl group. Instead you have this, right? When you are adding a new nucleotide, where do you add a new nucleotide? The hydroxyl group. If you don't have a hydroxyl group, can you add in a new nucleotide? No. So what we do is when we give AZT, we hope it gets incorporated where thymidine is supposed to get incorporated. So right here, right, A, if I'm building a new DNA complementary strand and I have A here, what do I need to put in? A T, right? Instead of putting in a T, you put in AZT, okay? You put in AZT right here. Can I add in another one to bind to this one? No. So you're preventing DNA replication. Is that a problem? Yeah. You can't make new DNA. So you can't divide and make a new cell because you can't make any DNA for it. No. So AZT is a base analog, okay? That's what a base analog is. Okay. No, you don't need to memorize all of the types of base analogs, but no, there's base analogs for each base, right? If we have, right, we have a base analog for guanine, cytosine, thymidine, and cytosine. So all four we have analogs for, okay? Acyclovir, right? We use this a lot for like herpes, right? These are very popular for treating different like herpes infections or HIV, right? Viral infections. But this is just to show you all the different options we have of base analogs. Okay. The only base analog, I care AZT, and then I might say, I probably won't ask you that. I'll probably just say AZT. And AZT is the only like specific one that I'll say like, if I will link you to know a specific base analog, it'll probably be AZT. Other than that, I just want you to know how a base analog works and when it works best. Yeah. So these are just two more examples. Okay? Okay, rifampin. Our base analogs, did they target DNA or did they target RNA? DNA. Okay, that's important. Okay. Rifampin targets RNA. Okay, RNA transcription. Okay. It's bactericidal. We treat this a lot with tuberculosis. Okay. That's pretty much all I care. The rest is a little extra information. quinolones. Quinolones and chloramphenicals. These guys are pretty cool, okay? Ciprofloxacin, you'll see on your plates. Ciprofloxacin specifically targets gyrase, okay? Gyrase is needed when? DNA replication, yes, okay? So, ciprofloxacin, you're gonna see it works really well on your plates, okay? What ciprofloxacin does is it targets gyrase. Other examples are norofloxacin, enotracin. I only care ciprofloxacin. The rest are like as good to know, but I will only ask you this one. Okay? Try to minimize all the antibiotics that we need to know. We use ciprofloxacin whenever we're dealing with multi-drug resistant strains, so like multi-drug resistant E. coli, Staph aureus, and these guys were developed. Here's a new relationship. Hybrid. Okay, here. Hybrids. Let me just show you the picture because this explains it very well. So here's the thing. Let's say you have a bacterial infection, okay? And you're gonna start with penicillin, right? That's just going down your train, okay? Let's say you start with penicillin, and let's say that your bacteria has beta-lactamase, okay? If it has beta-lactamase, what's it gonna do to the beta-lactam ring on penicillin? It's gonna cut it, right? Here's a hybrid, okay? It cuts it, right? What's gonna happen? Bacteria resistant, okay? Here's my penicillin, right? Here's my beta-lactam ring. We're gonna attach a quinolone to that ring. So, if you have a strain that has beta-lactamase, aka resistance to penicillin, it's gonna cut that ring, then your quinolone gets activated, right? It's kind of like setting a bomb off once you're already inside. Kind of like a Trojan horse, right? You guys know the Trojan horse story? Right? Okay.
It's like a virus. It's like a virus.
Yeah. I like to think about it as like the Trojan horse, okay? And what this is gonna do is to help reduce that multi-drug resistant, right? Or help reduce any possible resistance that can be made. Because if your bacteria is resistant to penicillin, we're still gonna kill it. So it's not like we created that environment where only the best survive. If they survive this, boom, this is gonna get you. Okay? So this is a way to try to reduce that. Okay? But when you give it this way, let's say that your bacteria doesn't have beta-lactamase. If it doesn't have beta-lactamase, that ring will never be cut and the quinolone will never be released. So you're not just exposing your bacteria to quinolone for fun. It's only activated if it's resistant to the penicillin. Okay? So it's only activated when the beta-lactam ring is cut. Yes?
Like really, it survives infection because it's never released, and then you have another infection. Will that still be good if that new infection activates like penicillin? Will it always be good?
No, you'll flush this out. This is just for the bacteria that you have in that moment. But the beauty is you're not exposing other bacteria to this if you don't need it, right? Which goes back to our story of you only want to give antibiotics when you need them, and you want to make sure you give a high enough dose for a long enough time that you kill everything, so you don't leave that like 1% that's the best. Okay? So that's the quinolone. So this is we like to give them in that hybrid, okay? This video is a summary of all of the modes of action that we just talked about, okay? So I think this is a nice video just to resume all the modes that we said, gonna play it for you.
Chemotherapeutic agents are antimicrobial drugs that fight disease by targeting a particular structural difference between human cells and those of the microbe. Because bacteria have many such differences, it's much easier to develop antibacterial drugs. Viruses depend on the host cell's machinery, so there are fewer differences to target. And eukaryotic pathogens, such as fungi and protozoa, have fewer differences from eukaryotic human cells, making it difficult to find drugs that have selective toxicity against these pathogens. Antibacterial drugs have several different modes of action. One of the ways in which antibacterial drugs can kill bacterial cells is to block the formation of bacterial cell walls. Normally, the peptidoglycan layers are held together with peptide crosslinks. Penicillin and its relatives block the formation of these peptide crosslinks. This weakens the cell wall, ultimately causing the cell to lyse. Antibacterial drugs such as polymyxin attach to phospholipids in the bacterial cytoplasmic membrane and interfere with its integrity. Eventually, the cell lyses. Because this mode of action also interferes with human cell membranes, this antibiotic is used only on the skin, where the outer layers of cells are already dead. Antibacterial drugs such as tetracycline, chloramphenicol, and streptomycin kill bacteria by inhibiting protein synthesis. Tetracyclines block the docking site of the transfer RNA, preventing amino acids from being added. Chloramphenicol prevents formation of peptide bonds between amino acids. Macrolides like erythromycin block the movement of messenger RNA through the ribosome. Aminoglycosides like streptomycin cause a change in the shape of the smaller 30S ribosome subunit, resulting in misreading of messenger RNA and insertion of incorrect amino acids. Bacteria have certain metabolic pathways that are not found in humans. One of these is the pathway for synthesis of folic acid, a coenzyme used in the synthesis of nucleic acids. Humans do not have such a pathway and require folic acid as a dietary supplement. One of the substrates in this pathway is para-aminobenzoic acid, or PABA. Antibiotics such as sulfonamides or sulfa drugs block a step in the folic acid synthesis pathway by acting as a competitive inhibitor to PABA. This prevents synthesis of folic acid and therefore synthesis of nucleic acids. The bacterium is unable to reproduce. Most organisms synthesize nucleic acids in a very similar manner, so it's much more difficult to find differences to target for these pathways. However, there is a group of synthetic drugs called quinolones and fluoroquinolones that block DNA gyrase in bacterial DNA. They have little effect on eukaryotic cells. Other drugs, such as rifampin, bind more readily to RNA polymerase in prokaryotes than eukaryotes and can be used to block transcription.
So, right, that was a summary. Everything we just talked about. Okay. And then, the only thing I didn't mention was for that sulfa pathway, that's a good drug because we don't make folic acid, right? Where do we get folic acid from? Diet. So we can target that enzyme that is involved in the pathway to make folic acid because we don't have that enzyme. So we're not affected in that way. Okay. Now we're gonna talk about resistance. This is your last column on your worksheet, okay? So there's quite a few ways, right? We're gonna learn how we're gonna throw an antibiotic at it, and then what the bacteria does to overcome that antibiotic, okay? Things like alteration of drug targets, okay? So we can change the way that the enzyme looks, the bacteria can change the way that an enzyme is structured, right? It can change if it changes its ribosome, can that antibiotic now target that same ribosome? No, because that one size fit, right? We can change alteration of the membrane, so we can change how permeable a membrane is. And if we have something like polymyxins, right, that normally insert into that cell membrane, if we change and make it more rigid, right, that's gonna make things more difficult to be inserted. Yes? I'll go back to that. I'll find it. Okay. Development of new enzymes, right? So things like beta-lactamase, that's completely new, right? Bacteria made a completely new enzyme to fight this off. We have alteration of metabolic pathways. So to combat like sulfa, what they do instead is some bacteria can say, okay, you're blocking my synthesis. Now I'm going to take in folic acid through a different mechanism using a different enzyme, or they can just take in folic acid from the environment, just like us. So then like, aha, you block this, now I'm doing it this way. Okay. Or you can have these pumps. Basically, they just pump the antibiotic right out. You throw it in, it pumps it out. Okay. And then biofilms, right? Biofilms in general, right? If you have that thick layer, is it harder to penetrate? Yes. Okay. So we'll watch this one at the end. Okay, so alteration of drug targets. So for example, if you are changing, for example, it targets the ribosome, right? We talked about different antibiotics that target the ribosome. If the bacteria alters the way or alters that 3D structure of the ribosome, will that antibiotic still be able to target that ribosome? No, because now it looks different, right? If it looks different, can't target it. If it doesn't target it, then it can't work, okay? This is how they developed resistance to erythromycin, okay? If you can't bind, you can't inhibit, okay? Alteration of membrane permeability. So what some bacterial cells can do is they can change their cell membrane to be a little bit more rigid, right? If it's more rigid, then it's harder for polymyxins to come into the cell membrane, right? That permeability, the ability to allow some things to come in or out, if we change it and we make it more difficult, right? More rigid, not as loose, then the polymyxins can't come in. If polymyxins can't come in, then you can't make the holes, right? So it doesn't work. Okay? And this is through like a mutation, right? Genetic mutation, and then it can change that membrane permeability. Okay? Development of new enzymes, right? This is beta-lactamase. This is a completely new enzyme that was made. Okay, this wasn't a previous enzyme that was altered. This is a brand new enzyme. Okay, what does beta-lactamase do? It cuts the beta-lactam ring. If it cuts the beta-lactam ring, then it inactivates any antibiotic that targets the tetrapeptide bond through the beta-lactam ring. Okay? Does that make sense? Yes, okay. Okay, alteration of existing enzymes. So we can have, right, here we had sulfa. Sulfa binds competitively. PABA can't bind, and now no folic acid, right? This bacteria can't replicate. Okay. Now you can have this enzyme, look, it used to look like a Pac-Man, and now it looks like this. So now sulfa doesn't fit. Actually, PABA fits better. Okay, so PABA fits better than sulfa. Okay. So if PABA fits better than sulfa, now PABA's gonna bind, and it's going to continue making folic acid, right? Inactivating or rendering resistance to sulfa, right? It doesn't work. Another thing is it can just completely bypass, right? So let's say it's going to now absorb folic acid from the environment the way we do. No need for folic acid synthesis. So if you're giving me sulfa, doesn't matter. Now I'm intaking my folic acid the way that we do. So boom, doesn't work. Okay. First line, second line, third line of drugs, okay? Let's say, so this is what we have seen in things like gonorrhea, okay? You give a sulfa, right? Patient comes in, you give them sulfa. Then there's a mutation, and it did, let's say alteration of that enzyme pathway, right? So now sulfa doesn't work. So you have a strain you have a gonorrhea, right, bacteria, that sulfa doesn't work, so it's resistant to sulfa, okay? Then you're like, okay, I'm gonna give penicillin, okay? You give penicillin, and then all of a sudden, oh, now it has a beta-lactam ring. So now you have a gonorrhea that is resistant to sulfa, and it's resistant to penicillin, right? On that same plasmid. And what can it do with that plasmid, right? Keep it for a new generation. It can also transfer that information, okay? So then I'm like, okay, penicillin doesn't work. Now I'm gonna give it streptomycin, okay? And then with streptomycin, I could potentially, right, there can be one mutation on that same plasmid that has resistance to streptomycin, okay? Then what am I gonna do? Give the next one, right? We do have some strains of gonorrhea, yeah. We got like 7 different antibiotic resistance on there, okay? That's kind of scary. So this is where we come up with first line, second line, third line. So that just means this is the first drug that we gave. It's resistant to it, so we gave it second line drug. If it's resistant, then we have to give a third line drug, right? And that's when maybe, you might have to whip out tetracycline, Okay. How can we limit resistance, right? General rule, high dose, long time. Make sure you kill everything, okay? The fittest do not survive, right? You want to give multiple antibiotics to try to overcome resistance. So the synergistic relationships, an additive, a hybrid, right? A good type of relationship. And then restrict antibiotics only when you need them, right? If they're not necessary, right? Let's say you have like traveler's diarrhea. Do you really need an antibiotic? No, it just needs to come out and just take some fluids, right? Get some electrolytes, get some water, hydrate. You don't really need an antibiotic for that. Okay. If you have like a low grade fever, right? Do you need the antibiotics? No. Unless it's high, then like, okay, you consult your doctor, right? But if it's just a tiny fever, it's good because that fever is going to kill everything. Because your body's going to get too high, it's going to inactivate the enzymes of whatever it is you have, and boom, you feel better. And you never expose it to anything. Okay? So this is showing you treating for a long enough time with the highest dose. Okay? So patient comes in, right? So within the patient, let's say this patient has E. coli. Okay? You have different levels of E. coli within you. You have some E. coli that's highly sensitive to your antibiotic. You have some E. coli that's intermediate, and you'll learn about intermediate when we look at our plates tomorrow. Intermediate means some works, but not all of it. So it's kind of like, it kind of works, but I can still grow. So you have some E. coli that's intermediate, and then you have some E. coli that's gonna be highly resistant. So that's can be like that 1% that's like holds on for DOI, okay? So let's say we're like, okay, in order to get rid of your bacterial infection, it's a 10-day treatment, right? It's a high dose for 10 days. So, day 3, who are you killing at day 3? You're killing all of your highly sensitive, and you're killing like half of your intermediate, okay? You're probably gonna start to feel a little bit better, okay? Day 6, you've killed all the highly sensitive, basically all of your intermediate, and then what can be left is just that 1%, that 1%. Are you gonna feel a hell of a lot better? Yes, you might not even feel anything, because 1% of these guys may not be enough to cause an infection, okay? But they are there, okay? So if I remove the antibiotic at day 6, what can this 1% do? Reproduce, right? Then eventually you're gonna have high enough numbers, maybe 2 weeks later, and then you come back in, and guess what? That 1%, highly resistant. Now that's gonna be a whole other scenario, right? Harder to kill, because all the ones that are left was there. Instead of you just continuing to take it for 4 more days, right? What happens at day 10? They're all dead, okay? So we wanna make sure that we educate everyone who's giving the antibiotics so that they know to explain this to your patients really well, right? And then provide that education to your parents, to your patients, okay? So that's number 1 reason why. And that's one way to reduce resistance, okay? Another way is, again, when you're giving combos, right? What's a good synergistic combo? Penicillin, streptomycin, okay? Penicillin and streptomycin work better together, right? We talked about this on Thursday. And you can have additives, right? Additive. What you don't want to give is an antagonistic, right? Who is our antagonistic that we're going to remember not to give? Yeah, tetracycline penicillin. Okay, please don't do that. Please don't do that. If someone prescribes you that, pay attention. I probably wouldn't trust them after that. Okay, but you pay attention too. If someone gives you that, you might be like, hold up. Do you know these mechanisms act? You don't want me to get better, huh? Yeah. Last one is limiting exposure. So I know we talked about only giving antibiotics when you truly need them. There's another way that you're exposed to antibiotics, right? Chicken, right? Different meat products, right? When they say like antibiotic free, most, if not all, types of like cattle or animals are injected with antibiotics, okay? Why are they injected with antibiotics? Because they don't want the animals to die. If they die, can you sell them? No. So they give them antibiotics so that they don't get sick and they don't die, and so that they make it to the market. But what happens is now you have animals with antibiotics that you then consume. So you can expose antibiotics to yourself by consuming products that are full of antibiotics. My dad's like obsessed with that. He's like super clean eating health person. He's like, that has antibiotics. All of that. Right? And then another example. So these animals, right, they're fed with antibiotics. And then they defecate. And then you have like crops in that same area. Similar to like E. coli outbreaks in produce, if you have feces that has antibiotics in it and you introduce that to plants, and then you cut those lettuce or strawberries or whatever, and then you sell it to the market, and then you eat that. So there's, I'm sure you watch that. But there's different ways that you can also be potentially exposed. So the more that you limit for yourself, the better, right? The one that we can really control is when you're consuming, and then also maybe like if you're looking at your labels, like by the one that says like no antibiotics, they probably give some level of antibiotics, but probably not a lot. I'm sure it's probably like if you give below this amount, you can claim no antibiotics. They'd be crazy. I feel like ain't no way, ain't no way. But better than it does not say antibiotic free, right? Because then I know it's pumped full of antibiotics. Yeah. So here's some food for thought. Can we continue to develop new drugs to combat rising drug resistance in pathogens? Will pathogens eventually beat us? So here's the thing. In order to develop a new antibiotic, so you find a new antibiotic, you put it in lab, you test it, you produce it, and finally, last step, administer to the public, that time frame, 10 years. Okay? What is the time frame of a bacteria once exposed to that antibiotic to become resistant to that antibiotic? 30 minutes, right? If it divides within that division, can there be a possibility that there's a mutation and it's resistant to the antibiotic? So all it needs is 30 minutes and it can potentially be resistant to the antibiotic. How long did it take us? 10 years. Okay. So, right? Are we gonna win? No. No, right? Maybe, right, we gotta invest in other options, right? That bacteriophage is looking pretty good right now, right? Yeah, that's pretty great. That's pretty crazy. Took us 10 years, 30 minutes, boom. Right? E. coli, 20 minutes. Right? So, it's some food for thought. Right? So we wanna just implement those practices to try to avoid having to introduce more antibiotics. Okay? Right? Bacteria was here before us, probably be here after us. Not today's problem, but maybe billions of years from now, it's gonna be someone's problem, right? So let's do what we can to limit that. Okay. What was the one that you were missing? I don't have the worksheet. What? Who do I hear about?
It's fluoroquinolones and quinolones?
They both work the same way.
Okay.
Who did I care about? If I can't remember, probably don't care about it. I don't see anything that starts with an L that's in quinolones. I care about ciprofloxacin. I'm gonna try to find what I was thinking about, but if I don't give it to you, I probably don't care about it. Yes.
Remember too, it says, if a patient is allergic to penicillin, you still want to treat it with the antibiotic when it could possibly use. Would I be able to put vancomycin?
Yes. You can also say cephalosporin or carbapenem first, and if those two don't work, then you can give vancomycin.
Are we assuming that we know it's bacteria?
Can you
A
repeat
patient
that?
isn't responding to vancomycin. What are the two scenarios that could
Okay, so if your patient's not responding to vancomycin, what could be happening in your patient, or what could be two possibilities? And you said one was,
It could not be bacteria, it could be something else, or.
That's
is that
true.
what- or
Is it only
antibiotic
target
resistance?
gram positive?
it mainly targets gram-positive, so those are two potential scenarios. You could have a bacteria that's not a gram-positive, so it's not working very well. Or two, it could not be a bacteria. If it's not a bacteria, is vancomycin gonna work? No. So those are two valid points.
Or resistant?
Or it's resistant, 3 points. Any other questions that we didn't target, or I didn't go over? No?
So the hybrid combo would be the cephalosporin and the other one?
Yes. What did ciprofloxacin target? Do you remember? Yes, aka DNA replication. We'll know that, specifically for that one would be DNA replication. The other one's RNA polymerase. If you're thinking of something that's both, then it would be nucleic acid.
Cause I don't know if it, because we don't have that L, I don't know if it's talking about these two, or
It's
the two
like
that
that.
we have.
I would know each one individually.
So that could,
So-
She's talking about this one, so it has to be the DNA replication, she's not talking about this one.
right, we don't have to put anything for this section.
She said she's not finding, she said she'll let us know.
Yes. See if I find it in my notes. If I don't, then you probably don't care about it. One less to know. Questions on any questions on the worksheet? We feel good about the worksheet. Okay, if I give you a worksheet, does that mean that you're gonna see it again? Right? On the quiz, if you didn't see it on the quiz, I don't know, I haven't made the quiz yet, but if you don't see it on the quiz, are you gonna see it on the exam? Yeah. Heck yeah. Yes, so, on Canvas, there's gonna be an assignment that says antibiotic worksheet. You can turn it in there for 2 points. It'll be due next Monday, so we finish it today. Questions? No? Tomorrow's day 2 of our unknown, so hopefully if you grew it, you'll get to gram stain it. Okay? That's exciting, right? It's like you're getting a patient, and you get to play like provider, right? I give you a patient, give you a scenario, here's your specimen, figure it out. It's like, it's guess who, right? Who did it?
For the metabolic pathways, for the different
They do target 2 different enzymes, but same pathway. Yeah. And then the question where it says, what is the difference between each, what I'm referring to also is what is the difference between each relationship? So what's the difference between synergistic and additive and hybrid? So know the differences between each relationship. Yeah? Yes.
Is this the last chapter for our next test? Or do we-
Yes, this is
Okay.
this is it. This is it for exam 2.
What about the quiz? Is this,
We should-
8, 9, and 10.
8, 9, and 10.
Yeah, cause it was.
Yup, 8, 9, 10.
hey, no, okay.
So what are we reviewing?
Only, right?
What are we reviewing on Wednesday?
Does it say review day?
I don't know.
If I put review day, it should be the day before. I do review days the day before, or, the day before your exams. So those are usually Tuesdays.
So next Tuesday after we take our quiz, is a review?
Let me So we didn't hit chapter 12 yet, right?
Oh.
We'll start chapter 12 next time.
And that's on example.
That's an exam 3.
Okay.
So we can check out like.
Yeah.
Where does it say review?
Nowhere.
That's why I was like, wait, what? Where's the review? Oh, so I was like,
Teresa's trying to help us out here.
are you talking about?
I'll be here Wednesday, but mentally not really.
She was manifesting an extra review.
Oh, I was like, huh? I was like, where's the review? I'm like, I usually put review day, like, oh, lab practical review. I will do that on Tuesday. So Tuesday will be, yes. So with the day that you have your quiz, we'll start with the quiz. Then we're gonna work on reading the results for lab 14, which is a microgen test. So we'll figure out that microgen test. Then you're gonna get to look at whatever it is that you're doing with your unknown. You guys are gonna be at different steps. Depending on what you have, you guys are gonna be in different spots. Then we'll end with review for the exam. That's Tuesday. Yeah. And then Wednesday, we'll be lecturing chapter 13. And then Thursday is the exam.
2 chapters.
There's
Yeah.
no chapter 11. So we just go to chapter 12.
So just 12 and 13.
Yeah, so we'll start talking about chapter 12 and chapter 13. I don't know if I'll be on chapter 13. I'm like one day off. I'm like a day off, but that's fine. That's fine. We'll catch up. Okay. But yeah, that cuts lecture for what you need to know. And then labs, it's going to be labs 8 through 13. 13 was what you're going to look at tomorrow. That's your antibiotics. So it's milk lab, transformation, disinfectants, and antibiotics. Yes, Betsy.
The micro project is due like the week of Halloween, right? So that, right? It's like in 3 weeks?
The micro project,
Or the,
case study 2,
no, not the case study, the micro project, like where you have to do like a model and a picture book?
that is due whenever.
Oh, okay, I understand.
That's due whenever. Tomorrow, if you wanted. That's due whenever. Okay. But you don't have to do tomorrow. My advice is once you're done with case study 2 and you're chilling with your unknown and you're just doing those tests, then start to like throw in some unknowns, right? Right? We can.
Didn't you say October was like the crazy?
I did tell you that, right? October, November is kind of busy because you have all these things piling up. So the best way that you can kind of spread some of these assignments out, the better. Okay. Yes.
Or is there another?
Yes, this is the last case study. Okay, we only do 2. Yep, no more case studies. That's it. Last presentation. Yeah.
Yeah, like stop recording.